Hi, everybody. My name is Mrs. Silverger, and I am recording this from sunny Honolulu, Hawaii. I'm really sorry that I couldn't be there live today. But I am the Associate Director of the Asia Center for the Advancement of Oceanography, and I'm an Associate Professor in the Department of Oceanography in the University of Hawaii at Manoa. I'm excited to talk to you today about biogeochemical cascades in coastal ecosystems. So my day essentially starts off with this sip of coffee. And this caffeine rushes into my system as this external resource that then fuels me to start responding to emails right away, which then often leads to me forming new collaborations all excitedly. And that often leads to me saying yes to opportunities all the way on the other side of the world, like here in London. And this ultimately either elevates or, depending on who you talk to, can bring down my productivity. But essentially, this external resource coming into my system creates this cascade of actions and reactions that lead to advancing my productivity. Now, granted, when that coffee is taken away, the exact opposite will happen. I will probably respond to emails much more slowly, say no to a lot more things, and reduce my productivity. And essentially, this new resource causes this whole cascade, which is really similar to what also happens in coastal ecosystems. So, for example, coastal ecosystems live in these highly variable areas where lots of interactions, biogeochemical, physical, and biological processes are happening all the time. So we see lots of variability in light, wave action, CO2, oxygen, pH, temperature, salinity, nutrients, and all of these factors all interact with each other to affect biogeochemical and biological processes. And when we're talking about variability in these systems, they're variable in many different ways. So, for example, these coastal ecosystems are often incredibly temporally variable, especially relative to the open ocean. So these are data on pH over 30-day time series that was collected by Hoffman et al. In 2011. And you can essentially see that this temporal variability in these upwelling, estuarine, porous, and calcareous environments are incredibly high relative to the open ocean. And this variability is driven by a combination of both physical and biological processes and can interact with those. So, for example, this temporal variability can also change over space. These are some data showing the relative frequency of temporal variability in pH and oxygen over a 30-meter transect. And you can see this on the order of meters, the relative importance of daily or weekly or hourly variability changes quite dramatically with just a really small space there. So, of course, in addition to that temporal variability, coastal systems have really high spatial variability as well. These are some data that are collected on the spatial variability in at key sites in Hawaii, in Honolulu Bay. To orient you, this is the coastline. In this case, the blues are lower pHs and the reds are higher pHs. And this is actually data collected along a gradient of where groundwater, that is fresh acidified water, is coming into the system, creating this really high variability on the order of meters, driven by a combination of the physical input of new pH to the system, but also the response to that through biological activity. So when we're talking about biological drivers of pH variability, we often think about photosynthesis and respiration as two of these dominant driving forces. So for example, during photosynthesis, CO2 is taken up out of the water column, oxygen is put back in, and through a chain of chemical reactions that increase the pH in overall water. Respiration does the exact opposite. These organisms take up oxygen, they put CO2 back into the water column, and through a chain of chemical reactions that decreases CO2 overall. And so you might imagine that in an area of a reef that's dominated with macroalgae, you're going to have really different pH environments and areas of the reef that are dominated by consumers. And of course, these biological processes not only drive the pH variability, but they can react to it as well. So, for example, so we had a coral reef that is filled with these producers and consumers. They photosynthesize and respire in the ways that I just explained. which ultimately changes the pH variability such that during the day when there's light, we tend to have higher pHs when there's more photosynthesis than at night, regardless of photosynthesis is essential. Now, this natural pH variability from these biological processes can in turn around and influence those same organisms again. So, for example, that pH variability can influence calcification and dissolution. Often we expect a positive relationship between pH and calcification, where there's higher pHs, we tend to have higher calcification rates. And when pH is lower, we tend to see increased dissolution. But of course, to make matters even more complicated, that calcification and dissolution is not just responding to pH, but it's altering it. And that photosynthesis and respiration is not just gathering pH, but it's responding to it as well. So in coastal ecosystems, we see these very complex biological feedback loops, where organisms are constantly driving and responding to the environmental variability. And then when you add new things to the system, like hallucinant inputs or external solutes or resources, such as nutrients like nitrogen and phosphorus, these may elevate photosynthesis and respiration, which ultimately increases the pH variability, and in turn can influence calcification and dissolution sending along. And if we alter the benthic community composition, we can also influence the overall net photosynthesis and respiration of the ecosystem, which in turn affects pH variability and calcification and dissolution. So these biogeochemical feedback loops are constantly living in this cycle in coastal ecosystems. But to add to that even more, in addition to these organisms cycling with this carbonate chemistry, we can have these biogeochemical cascades where we see not only do these processes influence, for example, nutrient uptake in the reef or changing dissolved organic matter, and then those processes further changing benthic community composition, or maybe the quadratic nature of the community. And the template of the physical, human, and ecological background influences the relationship between each of these processes in this ecosystem. So one of the things that I'm really interested in my research program is trying to understand what are the causes and consequences of biogeochemical cascades in coastal ecosystems. And so the way that I'm going to describe biogeochemical cascades is this idea of changing physical chemical parameters as mediated by biological processes. So this is an example here where, let's say, we might add new nutrients to a system. This may alter the community production in that system. That community production may then further alter the inorganic carbon flux, which may then affect community calcification. And together, the community production and calcification may in turn affect the organic carbon flux. And today I'm going to talk to you about two different examples where we looked at biogeochemical cascades in coastal ecosystems. The first one is going to be looking at the inputs of submarine groundwater discharge and reefs. And I know that was the main title of my original talk, but I decided to also add on some examples from intertidal ecosystems as well, looking at how altered community composition in these rocky intertidal systems also can create these biogeochemical cascades. So first, we're going to talk about the inputs of submarine groundwater discharge on coral reefs, and to see if they create biogeochemical cascades in its growth. So I think there was a talk right before me that was focused on submarine groundwater discharge, so I'm not going to get into this too much. But essentially, submarine groundwater discharge is this understudied natural gradient, but it is found literally all over the world. And it's defined as the flow of any water from the U.S. marine or composite bay that moves from the land through the marginal seabed and into the coastal ocean. And as you can see from this figure over here, that the equatorial regions have particularly high fluxes of groundwater, and even more so are regions that are high volcanic islands, like I'm living here in Hawaii, as well as where I live in the French Polynesia. So groundwater at these sites tends to be highly tidally driven. So during low tide, you tend to see these pushes of high fluxes of groundwater onto the reef in areas where there are groundwater seeps. And they can also be the largest source of new nutrients being flushed out. And they also have an incredible cultural and societal importance. And so, in where one of the places that I work in the area of French Polynesia, groundwater is called the source, and it is often used as sites with a lot of fishing. So this is a fishing shop that is placed right over a groundwater sweep at a site that I work in that area. It's also used even for laundry and for drinking water, and also for other types of applications. And of course, like everything else in the world right now, groundwater fluxes are changing as a result of sea level rise, climate change, and changing precipitation, also changing from coastal development as well. And they act as a conduit for anthropogenic pollutants because septic can seep into the groundwater and fertilizers can seep into the groundwater. So even though they have naturally high nutrient concentrations, they also can have anthropogenically elevated nutrient concentrations as well as other types of anthropogenic. But we don't really have a great understanding of the baseline effects of groundwater and soil biogeochemistry and ecology. So that brings me to what I'm going to talk to you about today, which is does submarine groundwater discharge create biogeochemical cascades in case of the reef? This is from a paper that was recently published in Ecological Monograph, and the QR code if you're interested in reading it. But we worked at two sites in the area, French Polynesia, which is in the South Pacific. The area is a high volcanic island. And we had these two sites that are on the western side of the island in the area. And at each of those field sites, we have 20 different experimental locations and one research point where we characterize the biogeochemistry of the water. So this is an example of one of those sites. This one is called Cabral, and it had cross-directional tidal flow moving inshore to offshore. Each of these yellow points was one of our reef sites, and this red point with the picture in it shows one of the research points that was on shore. And this water is so fresh that during low tide, essentially dogs would drop here and rinse directly from it before it was making its way out on the reef. This is also when there's spot showing, the speed of the flow, and we call this the flow site because this is actually really slow water movement along the reef. Then we worked at a site a little bit further south called Ferrari. This is a picture of that site. Again, those 20 locations along the reef with the seepage point. In this particular case, the water was highly unidirectional, always flowing in this direction over here, and was moving much more quickly, as you can see from the plot. So here we're going to call this the back site throughout this plot. And this is an image of what that subtidal sweep looks like. This fuzzy area over here is actually that fresh water that is being eluted out onto the reef with what we call the sled, which is measuring pH, oxygen, salinity, temperature, light, and a couple in depth and a couple of other parameters. So we did sampling at high tide and low tide, and during the day and night, during the wet season and the dry season, at all 42 locations, but at the seepage point, we also added a lot more points so we can get a better characterization of what is coming out of that groundwater seepage. So the background template at each of these sites is quite different. So again, we've got Cabral on the left, which is called the source site, and Ferrari on the right, which is called the fast site. And in addition to differences in the flow environment, there's also pretty big differences in the relative amount of nutrients that's getting out into the reef. So, for example, in the water collections right next to the sink, both of them had elevated nitrate plus nitrite, but at Cabral, it was 32 micromoles per liter, which is already extremely high. And at Ferrari, it was 280, which is totally wild and an incredibly high concentration of nitrogen for Ferrari. There's also differences in the relative human impact. So you can look from this aerial image. This is a single family home that owns pretty much the majority, if not all, of the nearby land, where at the fast site, there's more direct human influence. The majority of these homes are Airbnbs, with a couple of local homes as well, but you see a higher density of homes per square hectare here than you do over here. There's also pretty big differences in the income covenant chemistry between the two sites. So at the slow site, the water was highly depleted in alkalinity relative to incoming seawater, coming in around 1,500 kilograms. And at the fast site, it was extremely elevated, coming in at around 5,000 micromolars per kilogram. And also, there were differences in the pH in the groundwater relative to the seawater, but they were pretty similar between both the sites. In both cases, the groundwater coming in with the 7.2, and the background community composition was different between the two sites, but not really dramatically. So the slow site had about 20% soil cover, and the fast site had about 13% soil cover overall. So overall, we wanted to see does SUV-driven solute coming from the sea create biogeochemical cascades, and does the reef template affect these direct and indirect relationships between groundwater and reef metabolism? So to do that, we've created this hypothesized biogeochemical cascade using the Bayesian structural equation model. We have this system of 6 equations based on our current understanding of groundwater and coral reef biology. And a lot of the background information from the hypotheses came from a combination of our papers that we've already published here, but also, quite frankly, from a lot of the papers published by the people that are talking in these sessions today. So I'll walk you through these hypotheses first that we had. Our first hypothesis was that with increased groundwater, we see increases in the concentration of nitrate. Then with increases in the concentration of nitrate or other nutrients, we see elevated cycling of net ecosystem production. That elevated net ecosystem production then releases dissolved organic matter in the form of humus. And humic-like dissolved organic matter is associated with gridinous dissolved organic matter, also have been shown to be the primary exudates that come out of primary species. The epsilon stands for fluorescent dissolved organic matter. We also hypothesized that it would be elevated pH. That elevated pH we hypothesized would affect the net ecosystem classification. And then we hypothesized that net ecosystem classification would release proteinaceous dissolved organic matter, which is usually associated with animal or microbially driven dissolved organic matter. So what did we actually find? So the way that I'm going to show you the results here are in the solid lines. It'll be data that was collected from the dry season. And in the dashed lines, it's going to be data that was collected from the wet season. The blue-green lines are from the slow site, and the red lines are from the fast site. So our hypothesis was that groundwater, which we showed as a proxy for in this paper, was going to be positively correlated with the concentration of nutrients on the across those 20 sites, 20 locations per site. And we saw that at the fast site, a really strong positive relationship. And at the slow site, there was no relationship whatsoever between silicate and the nutrient concentration of the reef. Even though we saw really strong relationships between silicate and nutrients actually in the groundwater content. So this is really interesting because what this suggests is that at the slow site, because the water is essentially just sitting there, there's increased time for biological uptake. And so we're losing the ability to detect the relationships between silicate and nitrate on the reef because the organisms are taking up the nutrients relatively quickly. At the fast site, however, we see that strong relationship because the water is constantly being replenished and moving over the reef, and they don't have enough time to pick up that nutrient on the way. We then hypothesized that nitrate would be positively associated with net ecosystem production during the day, so elevated photosynthesis essentially, and negatively associated at night, which is elevated respiration, mostly due to microbial driven respiration, which is what our hypothesis was. And some of the time we saw that, we saw at least at both the sites and one of the seasons, we saw pretty strong relationships between the nutrients and the net ecosystem production, and then sometimes we didn't see a relationship at all. So what this shows is that there's this nonlinear relationship between nutrients and production that could be driven by a combination of whether or not the nutrients are saturated on the reef during that time, as well as changes in the background environmental conditions. So, for example, the flow or even the community composition of the reef. So maybe nutrients are saturated during the times when we saw this spot relationship, which might even elevate the productivity of the. We also hypothesized that net ecosystem production would be positively associated with the relative amount of humic dissolved organic matter in the reef. So this is commonly exudated from primary producers, and that is exactly what we saw. So pretty much all the time, we saw a pretty strong relationship between elevated net ecosystem production and elevated humic fluorescent dissolved organic matter. We also hypothesized that when there was increased net ecosystem productivity, there would also be increases in food, which is exactly what we saw, and a very strong relationship between production and food. Almost lastly, we hypothesized that pH would be positively correlated with net ecosystem calcification. And we saw that at the fast site, but we did not see a relationship between pH and energy at the flow site. And there's a couple of different reasons for this. Like, could be due to hysteresis in the in the time frame in which we were measuring pH and calcification, which was instantaneous, so we weren't able to test that. It also could be similar to the differences in the flow regimes that we tend to see higher calcification rates at sites where there's higher flow rates, so that they are more impacted by the solutes being delivered to them. And then lastly, we hypothesized that with elevated calcification, we would see elevated prognosis or animal or microbially driven dissolved organic matter. And interestingly, we saw the exact opposite of our hypothesis, which was pretty strong uptake with higher calcification. And so what this suggests is that there is a stronger consumption of coordination dissolved organic matter than the production of that prognosis is elevated. So essentially what we're seeing here is that the addition of this new solute from groundwater creates this chain reaction that scales all the way through the system and ultimately impacts ecosystem metabolism through production and calcification and the recycling of organic and inorganic carbon. And if we just look at direct relationships, we would lose our understanding of how each of these processes are interconnected. So does groundwater create a biogeochemical cascade? I would argue yes. That we do see that this new nutrient has a strong indirect effect, direct and indirect effect on the inorganic carbon and the organic carbon substance. So essentially, the study shows that groundwater is a widespread phenomenon, may be greatly important to ecology and oceanography of coastal ecosystems, and might be relatively underconsidered with deriving a lot of these important ecological processes. Groundwater could also use microbial loop with the augmented dissolved organic matter, both through direct delivery of groundwater, which we discussed in different papers, and also indirectly through the groundwater altered biology, which is then releasing or taking up dissolved organic matter in the water column. Lastly, or actually the second to last, the effects of groundwater on biogeochemical cascades varies with template of the reef and the composition of the groundwater. We did not see the same patterns at both of these sites. We saw two different patterns between those sites depending on whether it was at fast water moving reef, differences in the benthic composition potentially, and differences in the solute composition of the groundwater as well. And then lastly, we argue that this approach allows us to uncover the direct and indirect effects of groundwater on carbon fluxes, which is important when trying to consider how groundwater is cascading throughout the ecosystem and affecting the ecosystem overall. And very briefly, I just wanted to give you a little bit of a sneak peek into some of the other groundwater work that we've been doing, highlighting how groundwater affects coral growth and coral competition, which we have published by Carolyn et al., as well as looking at community function by Barnes et al. We've also done some work looking at benthic biodiversity, and we have a handful of studies that are in review and in prep, looking at these exact same sites how groundwater is affecting the water column microbes, for delivery, fish herbivory communities, and coral metabolism. So our goal is to try to create this holistic story of how groundwater influences individual ecosystem processes that all contribute to these carbon separation theories. And because I'm not there in person, like I often get this question from people that say, this is just fringing reefs in isolated locations. Does it even matter? And I would argue back, number one, that it's not just some fringing reefs. In fact, here's a video of me scuba diving right next to a groundwater sink that was like a fire hose explosion of groundwater on a coral reef in 40 feet of water. And that I think that groundwater is much stronger or much more important to driving processes on coral reefs than we may have previously thought with them. So transitioning over from looking at the interest of groundwater discharging coral reefs, please proceed biogeochemical cascades. I wanted to touch base a little bit also on how changing benthic community composition can also cause these biogeochemical cascades as well. And I'm using rocky or tidal ecosystems as my example there. So one of the reasons why I love working in rocky or tidal systems to answer these types of questions is because they're filled with tide pools, which are essentially these microcosms of the sea, where we can manipulate these tide pools independently, but they still persist in nature. And we can characterize the changes in the benthic community composition and the water column collagenous and microbial community composition. And we can measure biogeochemistry in those pools and relate everything to it. So in these next examples, I talk to you about how community composition affects pH variability in a natural system using these tide pools, these microcosms of the sea. And then we look to see if the loss of foundation species alters these biogeochemical cascades. And here we're just focusing on the inorganic carbon pool for this particular experiment. If you're interested in the organic carbon pool, we've got a study that we just submitted into review that I'd be happy to talk to you about Okay, so first, we changes in community composition affect pH variability. So this was done in about 57 tide pools across the whole west coast of the U.S., ranging from Southern California and San Del Mar to Central California and Monterey and Bodega Bay to Bob Creek up in Central Oregon, and we worked at about 13, 15 tide pools at each location, where we characterized the benthic community composition in the pools and different aspects of the biogeochemistry with the focus here that I'm going to talk to you about today on pH. So here we're going from south to north. We're going to have daytime measurements on the left and nighttime measurements on the right. And these red lines show the adjacent ocean sample that we collected to kind of show the background conditions. And so as you can see, there's nothing much exciting going on here yet. But when we overlay the data from the tide pools, each of those lines represents one tide pool, we see dramatic pH variability. In fact, at some of these sites, you see pH ranging from 7 to 9 in 6 hours. A 2 pH unit change in just 6 hours in these tide pools, and there are a ton of organisms, even of calcifying organisms living in these pools just happily. So just to put this into perspective, the global oceans are supposed to change by around 0.1 pH units in 100 years, 50 to 100 years, and this is 2 pH units in 6 hours. So pretty wild pH variability in the pools. And so, of course, the question is, what is driving this variability? So we looked at 3 different methods of variability for this paper. We looked at the range, the mean, and the deviation from the ocean. Just going to show you the results for the range and the mean right now. We compared a handful of different biological and physical drivers, including producer dominance, which was the relative amount of fleshy macroalgae minus consumers, so like mussels, bronchics, for example. We looked at ecosystem metabolism through PA and VOC scopes, looking at this essentially the relative amount of calcification during the production. We looked at the pool size. We looked at the pool location in the interpetal. We looked at an integrated measure of light, and we looked at the temperature range over the sampling period. So right now, these are standardized effect sizes for the range model. where if those lines overlap this zero line, that means it's not statistically significant, and if they're different than that line, it means it is. And so the first thing that you see is that the biological drivers substantially outweigh the physical drivers of case variability in these tide pools. Even though we had tide pools that range in size from like buckets to like pools. But the biology that was in there and the composition of the biology seems to be way more important driving that case variability. When looking at the mean, we also included interaction between day and night. Again, the biological drivers were the only thing that came out as statistically significant. So here's what this looks like graphically. So on the x-axis, we have producer dominance, where the negative values mean that they are dominated by consumers, like mussels, and the positive values meant that they are dominated by producers, like macroalgae, and then you have the range in pH on the y. And we saw across all 57 tide pools this really strong positive relationship, regardless of where it existed on the west coast of the year. And when looking at the mean, we interestingly saw that during the day, there was a strong positive relationship between mean pH and tide pool community composition. And at night, there was no relationship at all, which is surprising because I thought there was going to be this like mirroring where you would see this stronger decrease in pH where you also had this increase in producer dominance because they also aspire. But what we think is happening is that at night, the pools get pretty hypoxic and oxygen limited. So it's essentially limiting the amount of respiration and the amount of pH that can drop. So during the day, we're seeing increases in photosynthesis, and at night is oxygen is limiting respiration and limiting how much the pH can drop. So essentially, composition has substantial control over pH variability, and shifts in community composition will alter these pH dynamics. So if we change that community composition, then is it going to create a biogeochemical cascade within the energetic carbon field? So to do that, we focused in on that Oregon site, where we focused on two dominant foundation species that exist out there, which are the mussels, Middle Californianus, and surfshark, which is south there. And in general, both of these foundation species are changing quite rapidly all over the world. In Southern California alone, mussel loss has changed by about 40% since the 1980s, essentially. And globally, seagrasses have declined by around 30%. So these foundation species are disappearing rapidly, and they're happening, over this climate change time scale, but also quite rapidly because of heat waves. There was a massive heat wave in Northern California, Southern Oregon, just a handful of years ago that killed something like 1 billion mussels in a couple of days. And we also see bleaching of surfsharks and seagrasses from this heat wave as well. And so is this going to have a scale up to affect the carbon footprint of these ecosystems? So what are the direct and indirect effects of these foundation species loss on ecosystem functioning? So this project was led by my grad student, Jennifer Fields, who is now a program manager out in California. And she essentially worked at these tide pools that were either dominated by mussels or dominated by surfsharks, and ran a before-after control impact design where she measured a bunch of biogeochemical parameters, removed all the foundation species and half the pools, and then measured these parameters again. So dial changes in temperature, light, nutrients, pH, community composition, that you just success and calcification. And then removed from half the pools and she did it again. So what she essentially found was that the renewal, of course, had a huge impact on the chemistry of the tide pools. So this is a principal components analysis. These dots, each dot represents an individual tide pool. The open circles represent the tide pools in the before period, which was July, and the closed circles represent the tide pools in the after period, which is August. These lines represent the median of the control, the ocean, and the removal. And so you can see this is the direction between how the biogeochemistry of the background ocean changed from July to August. And the control tide pools essentially fully mirrored what the background ocean conditions were doing. And the removal pools shot off into a completely different direction and had completely different biogeochemical environments. This is the biplot showing where each of those parameters lie in two-dimensional space. So this is for the surf grasses, this is for the mussels, this is the ocean sample for the mussels, and here is the control for the mussel again. The axis is moving in the exact same direction. However, when the removal tools are considered, you see that the biogeochemistry is changing dramatically in a different direction. So the short answer is, of course, the removal of these organisms is having a huge impact on the biogeochemical environment. In addition to the biogeochemistry, it also influences the temperature quite dramatically. So when we look at foundation species loss in the Thalassodus, the surf grasses, we actually saw on average an increase in 7 degrees Fahrenheit or 3.8 degrees Celsius in the tide pools that had the Thalassodus removed. And similarly in the mussels, though not as dramatically, we saw an increase in about 1.3 C or 2.5 degrees Fahrenheit. So this is a pretty massive increase in temperature in tide pools. And as we know, temperature can also influence metabolism. So our questions are, how do these changes in chemistry and temperature scale up to affect the tide pool ecosystem? So again, we use the structural equation framework, where we looked at the differences between the before and after removal periods. And we started with how much foundation species loss and important covariates like tide height and size of the pool influenced net ecosystem production and calcification as mediated by changes in light and temperature and nutrients, changes in the micro and macroalgal cover, as well as pH. So I'm going to show you here, again, the structural equation model. I'm only going to show you areas for the significant effects that were retained in the model. Blue arrows are positive relationships, negative arrows are red arrows are negative relationships. And of course, all these hypotheses are built off of what we know about the biology of how these ecosystems work. So we essentially saw that with surf grass loss, we saw this increase in temperature, this increase in micro and macroalgal cover, and that those changes in that change in temperature ultimately increased net ecosystem production. We also saw that surf grass loss increased pH, and that increase in pH influenced net ecosystem classification. So essentially, we saw these indirect effects of surf grass loss on ecosystem metabolism, mostly via changes in the temperature and also the light environment as well. When I just looked at the relationship between surf grass loss and net production and calcification, we didn't see a relationship. We only saw a relationship when we considered these mediating factors in this process. Similarly, when looking at the mussels, we saw that mussel loss increased micro and macroalgal cover and pH. That increases in micro and macroalgal cover increase net ecosystem production, and all of that influenced pH. And here we saw that mussel loss indirectly influenced net ecosystem production, mostly through changes in the microalgal cover that increased dramatically after mussels were removed from their system. Again, if we looked at direct relationship between mussel loss and NAP, we lose that relationship. We see it through these mediating factors. So ultimately, we saw that species loss not only affects biodiversity, but it stimulates these biogeochemical cascades in these coastal ecosystems. So community composition has substantial control over pH variability. The loss of foundation species indirectly affected ecosystem metabolism by altering the physical chemical environment. We saw that the loss of foundation species could stimulate these biogeochemical cascades where we remove one species from the ecosystem and it changes the carbon flux in that system, and it scales up. And in fact, we also have another study that I mentioned earlier where we're looking at the organic carbon as well as the pelagic microbial community, and this a little sneak peek, it affects that too. The loss of macrophytes could confound the effects of ocean acidification, essentially, because if they're losing these producers that increase pH in these coastal ecosystems, then we can have an even stronger negative impact on ocean acidification, on, for example, calcifying organisms that are particularly dependent on the pH environment. And then lastly, this also shows that this biogeochemical cascade framework is really applicable to all coastal ecosystems. So what we talked about today was what are the causes and consequences of biogeochemical cascades in the coastal ecosystems. There's focused on the organic and inorganic carbon sectors. And we talked about the inputs of submarine groundwater discharge on coral reefs, as well as altered community composition in the carbon ecosystem. And if you only walk away with one thing from this talk, I want you to walk away with this one message. Of course, this idea that every single thing is connected, you add or remove one thing to the system, and it just causes this cascade, this chain reaction of different processes, and that also using a biogeochemical cascade approach really allows us to uncover both the direct and indirect drivers of ecosystem processes. And this really reinvigorates classical frameworks in ecology and oceanography by Odom and Odom, for example, that connects ecosystems to communities. So thank you so much for listening to my talk. Of course, I have to thank all of my funding sources, my amazing collaborators on all of the papers that we talked about today, as well as all the student contributions that helped out with those research projects. Now, I'm sorry that I'm not there live to answer any questions, but please don't hesitate to email me at silvergray@hawaii.edu if you have any questions. And I look forward to hearing from you. Again, thank you so much for listening to me.